Citric acid cycle is also known as the TCA cycle, tricarboxylic acid cycle, and it's also known as the Kreb cycle. So those three terms or names for the metabolic pathway are synonymous. Now the TCA cycle is an actual metabolic circuit. It consists of 8 reactions that go around in a circle. So there are ways to actually memorize the intermediates in the citric acid cycle. One that I like to use is this mnemonic. It is can I keep selling shirts for money outside. The CAN stands for citrate, the I stands for isocitrate, the keep stands for alpha ketoglutarate, the SN stands for succinyl-CoA, the S stands for succinate, F stands for fumarate, M is for malate, and then from malate we go to O, which stands for oxaloacetate. Now you have to remember that the oxaloacetate goes back to create this circle. This is a metabolic circuit, and it reacts with acetyl-CoA. So acetyl-CoA actually feeds in to this TCA cycle, and oxaloacetate reacting back with acetyl-CoA regenerates the start of this cycle. If you remember, the acetyl-CoA is generated by PDH multi-enzyme complex, and now you have a cyclical metabolic pathway. The TCA cycle is actually housed in the mitochondrial matrix. So organisms that house a mitochondria within the double membrane of the mitochondria inside of the innermost region, in higher eukaryotes, the TCA cycle or the citric acid cycle or the Kreb cycle is sustained. So let's go over the individual reactions that constitute the TCA cycle. Before we do that, let's just do a balance sheet of where we are at energetically speaking. If you remember in glycolysis, the first step of glycolysis, hexokinase, and the third rate-limiting enzyme of glycolysis, catalyzed by PFK1, those take in ATPs. So the cell has to dedicate ATP for those steps in glycolysis. However, you actually generate 4 ATPs via substrate level phosphorylation. You generate that by the phosphoglycerate kinase step and also the pyruvate kinase step. That's the terminal step in the 10-step pathway known as glycolysis. So that is 4 ATPs. You may be wondering why this is a 4. It's because we double everything up in the triose phosphate isomerase enzymatic step. So after that enzyme catalyzed reaction, everything gets doubled up. So we have 4 ATPs that are credited via substrate level phosphorylation, 2 that have to get deducted when ATP is dedicated in the hexokinase and the PFK1 enzymatic reactions. So the net is 2 ATPs. We also get 2 reducing equivalents in the form of NADH. Those are going to be plenty useful when we talk about the electron transport chain. So you may know that ATP is a good thing to have, but these reducing equivalents, NADH and FADH2, are just as vital. We also get another 2 NADHs from the PDH multi-enzyme complex. So there's a video early on that goes through the steps in that catalysis that takes pyruvate generated in glycolysis and converts it to acetyl-CoA. It's a super enzyme, consists of E1, E2, and E3, and has about 5 essential cofactors that catalyze that reaction. One of the products of the PDH multi-enzyme complex is also 2 molecules of these reducing equivalents in the form of NADH. Finally, the reaction that we're going to go over now also generates a lot of reducing equivalents. It generates 6 NADHs and 2 FADH2s, the flavin adenine dinucleotides. So these reducing equivalents are going to be a reservoir for lots of energy when these NADHs and FADH2s give up those electrons, and those electrons go to the electron transport chain. You also generate energy in the form of GTP in the TCA cycle. So overall, for higher eukaryotes or advanced aerobes that have the capability to perform the TCA cycle, there's the potential for a lot of energy, not just in the form of ATP and GTP, but energy that can be utilized and captured in the form of donating those electrons to oxygen and coupling that with ATP synthesis. So we want to contrast substrate level phosphorylation with oxidative phosphorylation. That's another way to generate lots of ATPs through these reducing equivalents. The first step in the TCA cycle is catalyzed by the enzyme citrate synthase. So here we have oxaloacetate. Oxaloacetate is going to react with this high energy molecule known as acetyl-CoA. That coenzyme A is a very good cofactor, but it's also a high energy molecule. In the next slide, you'll see a structure of coenzyme A. Usually we just abbreviate it in this manner to avoid its complicated structure. So the coenzyme A is a good leaving group, and that's going to come off as CoAsh and get recycled back. So in this reaction, these two carbons, the CH3 and the C double bond O, essentially go to one of the carbons in oxaloacetate to give you the 6-carbon molecule citrate. The mechanism for citrate synthase involves an essential hydrogen bond between the histidine and the C double bond O of acetyl-CoA. And then another active site residue is aspartic acid in its aspartate form, where this deprotonated oxygen picks up or abstracts a proton from acetyl-CoA. So this very strong enol actually does nucleophilic attack on the oxaloacetate. As a net effect, we have a 6-carbon molecule known as citrate. Here you can see the carbons from CH3 and C double bond O going into this carbon here of citrate. So just as a reminder, here is the structure of coenzyme A. This is the first reaction of the TCA cycle, but you can see here that the coenzyme A actually is a very complicated molecule. And really all these carbon atoms that are essentially going to be transferred are going to be conjugated to this thiol group. So as an example, you can see here, this thiol group is going to be the site of attachment. Take a look, for example, acetyl-CoA or succinyl-CoA or propionyl-CoA. So just as an abbreviation, we put coenzyme A as CoA, but really the actual structure is very complicated. The second enzyme in the TCA cycle is aconitase. Aconitase does essentially an isomerization of citrate to isocitrate. So here you see these two this hydroxyl and this hydrogen sort of switch places. So the structure is exactly the same, just the arrangement of the atoms have changed. And so this is the enzyme that actually contains, an iron sulfur cluster that participates in the mechanism. And iron sulfur clusters are important in not just the TCA cycle, but in the overall iron homeostasis of the body. So there's a lot of literature that talks about iron and iron dysregulation in Alzheimer's disease and hemochromatosis, in which there's iron dysfunction in the blood. Also, hemoglobin contains iron. So there's a whole biochemistry around iron. And aconitase that catalyzes the second reaction in the TCA cycle also contains these essential iron-sulfur clusters. The enzyme is involved in a reversible dehydration and hydration reactions. And clinically, besides many other things iron is involved in, aconitase is implicated in a disease known as Friedreich's ataxia that involves difficulties in gait. So mutations in this enzyme have been found clinically in this disease. Also take note of the arrows. Reaction 1 and reaction 2 are reactions in the TCA cycle that are in equilibrium. So that's something you kind of want to take into consideration when you look more closely at each of these 8 reactions of this cycle. The third reaction of this cycle is catalyzed by isocitrate dehydrogenase. And if you notice here, the reaction just goes in one direction. There is no real equilibrium that's established. So enzymes that go in one direction and do not have a reverse counterpart usually are enzymes that are very regulated by allostery in the pathway, or they could be a rate determining step in a multi-step metabolic reaction. So isocitrate dehydrogenase converts isocitrate to alpha-ketoglutarate. So if you take a look at what essentially is happening in terms of the atoms, this OH group is going to get oxidized to the C double bond O, and simultaneously to that, this NAD plus is going to get reduced to the NADH. Notice that this COOH stays here, and this COO from the carboxylate gets decarboxylated to CO2. And then finally, a very good thing here, as the NAD plus gets reduced to the NADH. So not only is this an oxidation reduction reaction, but this is also a decarboxylation reaction because this COO minus comes off as CO2. So one good thing about this reaction is that it produces NADH. That's something you really want to consider as being favorable towards the cell, something that contributes towards the reducing pool or a reducing equivalent pool inside the cell. The reaction is irreversible. It goes in one direction. So that could be a site for regulation. And this is an oxidative decarboxylation reaction. So just a word here about the use of these protons. So here I had the proton, here I have the proton removed. It's really not of consequence. It's just that sometimes these carboxylic acids and carboxylates have a pKa where the proton can come off and on. So not have to get so confused about the addition of this proton here versus the proton not being there. It's really a function of the pH that the cell resides in, and it's also a function of the pKa. So isocitrate, if I fully protonate it all, it will be isocitric acid. And alpha-ketoglutarate, if I protonate it all, it's going to be alpha-ketoglutamic acid. So generally, within the physiologic pH of the cell, these protons come off, and alpha-ketoglutarate has the protons removed on the carboxylate ends. Another important thing about alpha-ketoglutarate is that alpha-ketoglutarate can actually get transaminated to its corresponding amino acid. So that corresponding amino acid is called glutamate or glutamic acid if you fully protonate the molecule. Transaminases are enzymes that are ubiquitous in the liver, and they play a very important role in switching between what is known as the alpha-keto acid and the alpha amino acid. Now, you know there's 20 alpha amino acids, but these alpha amino acids can switch to their alpha keto acids. You can see here, here's the keto group. If I amidate that, I'll have an alpha amino group and essentially an alpha amino acids. So I put that bullet point here just for students to realize that you actually have a point where you can shunt alpha-ketoglutarate away from this pathway towards the production of an amino acid. Likewise, if the cell needs to undergo more flux towards the TCA cycle, you can take glutamic acid or glutamate, transaminate it, and you can have alpha-ketoglutarate formed, and that alpha-ketoglutarate can then feed into this step of the TCA cycle. So from the TCA cycle, you can actually take intermediates out and use that to contribute to the amino acid pool via these enzymes known as transaminases. We'll study transaminases a little bit later on in the course towards the end if time permits. But these are very powerful enzymes, and they're clinically relevant, particularly with regards to heart attacks, any type of myocardial infarction, any kind of ischemic or stroke event. The liver, for whatever reason, leaks these transaminases out into the blood plasma. So there's a lot of clinical interest in these transaminases. But for now, I just want to illustrate the fact that you have the capability to generate amino acids from TCA cycle intermediates. It goes back to flux and shunting in and shunting out, which is a major theme of metabolism. This enzyme requires the metals magnesium and manganese. So if you ever wonder why you need magnesium and manganese in your body, it's simply because of the fact that they help this enzyme function. The magnesium generally chelates negative charges, and in this case for isocitrate dehydrogenase, the manganese also chelates some of these negative charges and negative polarities coming from the oxygen. Reaction 4 of the TCA cycle is alpha-ketoglutarate dehydrogenase. So anytime you see these dehydrogenases, you have to really think oxidation reduction. And for the most part, what gets reduced is NAD plus to NADHs. Not all the time, but a majority of the time the dehydrogenases generate NADH. So what is happening here? In this reaction, we have this terminal COO minus get decarboxylated to CO2, so it's an oxidative decarboxylation reaction. Coenzyme A actually gets conjugated to give you this high energy molecule known as succinyl-CoA. So we saw the structure of coenzyme A previously. It's this long, complicated molecule, but we're really only interested in that thiol portion, the SH portion of that molecule that sort of brings in carbon, or a collection of carbon atoms. In this case, it's bringing in succinyl, and it's being sort of hopped on this coenzyme A bandwagon. And also another very, very good thing is that we produce NADH. So this is the second NADH that's actually produced in the TCA cycle. So the enzyme goes in one direction. So keep track of the arrows, whether the reaction is in equilibrium or the reaction is just irreversible and sort of committed towards the products. As we stated, the reaction is one-sided, and this is an oxidative decarboxylation, and you're priming the molecule with coenzyme A. So we can state that succinyl-CoA is a high-energy molecule. That coenzyme A is a good leaving group. And a couple of words on this enzyme. This is the TCA cycle form of the enzyme that's housed in the mitochondrial matrix, but there's an isoform of the enzyme that is exactly involved in a different form of metabolism that is involved in the metabolism or the breakdown of branched chain amino acids like your leucine, isoleucine, and valine. Those amino acid side chains that are branched. Alpha-ketoglutarate dehydrogenase, a separate form of that enzyme, is involved in breaking down those amino acids. And just like PDH multi-enzyme complex, that version of alpha-ketoglutarate dehydrogenase has an E1 form, E2 form, and an E3 form. So if you remember, E1 has TPP, E2 has lipoic acid, and E3 has FAD, which gets converted to FADH2. So it's a superenzyme or multi-enzyme complex that is almost totally analogous to the PDH multi-enzyme complex. Henceforth, the mechanism and everything is also going to be the same. There's a clinical relevance of alpha-ketoglutarate dehydrogenase. It's involved in maple syrup urine disease. So if your body cannot metabolize these leucine, valine, isoleucine, these branched amino acid compounds, if they're in high amounts in your diet, if there's a dysfunction in that particular enzyme, the liver cannot process it functionally, and in essence, you get maple syrup urine disease. We study these very strange diseases in a higher level biochemistry class. We won't talk about it here in Biochemistry 1. All right, so reaction 5 of the TCA cycle is a synthetase, succinyl-CoA, that we generated from reaction 4 is going to do what is expected to do, and that is that coenzyme A is going to leave. Okay, so coenzyme A does what leaving groups do, and they leave. So coenzyme A leaves as CoAsh, and succinyl-CoA becomes succinate. Now, high-energy molecules such as succinyl-CoA are very exergonic. When that leaving group leaves, it releases a lot of energy, and that energy comes in the form of GTP. So a very good thing now. We've had 3 successive reactions where we produce reducing equivalents in the form of NADH, and here we produce GTP via substrate level phosphorylation. So energy that could be utilized that comes in the form of GTP. The reaction is reversible, so it is in an equilibrium, so it can go from reactant side to product side based upon the concentrations of either substances. So you have to think of Le Chatelier's principle here when anytime you're thinking about reactions that have directions both ways or reactions that are in equilibrium. And coenzyme A is a good leaving group. In terms of the mechanism for this reaction proceeds via a 3-phosphorylated histidine intermediate. So there's an enzyme in glycolysis, actually, 3-phosphoglycerate mutase, that also proceeds via a histidine phosphorylated intermediate. So this is basically a phosphorylation exchange. The phosphate goes to histidine, and then, which is part of the active site of this enzyme. And then the phosphate displaces the coenzyme A, and then GDP comes along and displaces the phosphate. So it's a multi-step mechanism. The active site residue is not histidine, but a phosphorylated histidine. In 6, we have 8 reactions in this cyclical pathway, and that takes the succinate and converts it to fumarate. So if you actually look at the atoms here, this CH2CH2 gets dehydrated, and now you have a double bond here, CH to CH. Where do those hydrides go? They actually go to FAD plus and reduce that FAD plus to FADH2. So succinate gets generated to fumarate. The enzyme that catalyzes this is succinate dehydrogenase. A couple of things about succinate dehydrogenase that is important. Number one, we make FADH2, so we make a reducing equivalent, a very good thing for the cell, especially if it's an aerobe and it's going to perform the electron transport chain. The reaction can go back and forth. It is in an equilibrium. And this is simply a dehydration reaction where the hydrides, the hydrogen and the hydrogen, just go to reduce FAD+. Succinate dehydrogenase is important because it actually feeds right into the electron transport chain. It feeds into complex 2 of the electron transport chain and is the only TCA cycle enzyme that is actually membrane bound. So the electron transport chain, which is really the next step in our sequence of studying metabolism as to what's going to happen to all these reducing equivalents. The electron transport chain actually is a series of membrane complexes. Well, here is an enzyme that's part of a metabolic pathway, the Kreb cycle, that actually is bound to the membrane. So it has a direct line to the electron transport chain. So the important thing to remember about succinate dehydrogenase is that it is the only membrane-bound TCA cycle enzyme. It is bound in the inner mitochondrial membrane, and that FADH2 has a direct conduit to complex 2 of the electron transport chain. We are at the second to last reaction in the TCA cycle, and that enzyme that catalyzes this reaction is fumarase, that takes fumarate and then sort of hydrates it. So this is a reversible hydration or dehydration. You can see what's happening to the atoms here is that this CH is going to get hydroxylated, and as a result of that hydroxylation, one of the protons goes to this carbon here, and that produces malate. So malate is important because it actually is involved in the malate aspartate shuttle. That's one way to get reducing equivalents into the mitochondria. The malate aspartate shuttle really is going to be a way that we tie together the NADH produced from glycolysis into the TCA cycle so that the NADH produced from the cytoplasm via glycolysis can go into the mitochondrial matrix and donate to the electron transport chain. But if you just look at the carbons there, it's a hydroxylation of fumarate to malate. The last step of this process is converting malate to oxaloacetate. So here, malate dehydrogenase, here's that word again, dehydrogenase. So think oxidation reduction. And for the most part, think of NADH being formed, or NAD plus being reduced to NADH. So here's the oxidation. This OH is going to get oxidized to this C double bond O. We produce NADH, which is a good thing. By my estimate, this is probably the 4th NADH that is produced in the pathway, or I think 3rd. This is the 3rd NADH produced in the pathway. And that's a good thing. It's producing reducing equivalents. Now, there's 2 forms of malate dehydrogenase. There's a form that's involved in the mitochondrial matrix. The one that's in the mitochondrial matrix actually goes in this direction towards the production of oxaloacetate. We also have a cytoplasmic form. The cytoplasmic form actually goes in the opposite direction. It actually goes towards the production of malate. So different isoforms housed in different geographic locations within the cell, and each version has its equilibrium shifted towards one form or the other. In the mitochondrial matrix form, the one that is involved in the TCA cycle, MDH1, malate dehydrogenase 1, favors the formation of oxaloacetate. That's just how the equilibrium is favored. And in the cytoplasmic form, the malate dehydrogenase favors the formation of malate and NAD+. So this MDH2 and MDH1 actually work in synergy with the malate aspartate shuttle in getting reducing equivalents into and out of the mitochondrial matrix, specifically NADH. And finally, oxaloacetate can be transaminated to the amino acid aspartic acid, also known as aspartate. So aspartic acid has all the protons in, aspartate has the proton removed from the side chain. And this is the similar situation that we saw with alpha ketoglutarate, where alpha ketoglutarate is the alpha keto acid that gets converted to the alpha amino acid glutamate. Here, the alpha keto acid, oxaloacetate is your alpha keto acid, can get transaminated to the alpha amino acid. In that case, that would be aspartate. So there's these transaminases again that are going to allow for intermediates to get shunted out if the cell so deems requires amino acids. Or if the amino acid pool is very high in the cell, those amino acids can get transaminated back to the alpha keto acid, in this case oxaloacetate, and then oxaloacetate can then feed in at this step of the TCA cycle. So you have pathways within the TCA cycle that can get shunted to make amino acids if amino acids are very low. And you have these transaminases that can do the opposite. If amino acid pools are very high, you can actually convert them into intermediates that get funneled into the TCA cycle and increase flux into the TCA cycle. So that is the 8 steps of the TCA cycle. Remember, this goes in a complete circle, so it's a metabolic circuit. A couple of things that you want to just keep in mind when you look at the reactions of the TCA cycle is reactions that produce reducing equivalents. So here we have reactions that produce NADH or its counterpart FADH2. Those are going to be pivotal because those will donate its electrons to the electron transport chain. Also, you want to be aware of reactions that produce energy in the form of substrate level phosphorylation. We contrast substrate level phosphorylation with oxidative phosphorylation. Now that GTP is essentially equivalent to ATP. We also want to take into consideration those reactions that only go in one direction. And in the TCA cycle, we have 2 reactions that actually are irreversible. One is isocitrate dehydrogenase, and the other one is alpha ketoglutarate dehydrogenase. So those reactions that are in one direction could be the site for allosteric regulation, and they could also be the site where you cannot go back. So those would be what we essentially call the committed steps in the TCA cycle pathway. And then finally, we want to also start thinking about shunting. So we discussed 2 examples of where intermediates of the TCA cycle, such as alpha ketoglutarate and oxaloacetate, can actually get converted to their corresponding amino acids. So that's very important because one of the main themes in metabolism is metabolites that can shunt in and shunt out. Many things that can get shunted out, for example, citrates can be shunted out and be used to make cholesterol, a steroid. Here we have fumarate. During metabolism of the amino acid phenylalanine, one of the intermediates that is formed in that multi-step breakdown of phenylalanine is fumarate. And where can that fumarate go? Well, that fumarate could feed in right here. That fumarate can feed in right here at this step of the TCA cycle. So it's like a contrast scan on your TV. You can have it go very high or very low. And shunting, feeding in and feeding out, sort of helps that process occur. And it also makes this TCA cycle more responsive to outside conditions in the cell. Don't forget, reactions that go in one direction, such as these reactions, reaction 3 and reaction 4. Don't forget that reactions that go in one direction can also be a site for regulation. So that's another layer of control that can regulate flux into this metabolic pathway. So it's very responsive to the outside needs of the cell based upon multiple modalities. One is it's responsive to amino acid content through these transaminations. It's responsive by allosteric regulation through these irreversible reactions as catalyzed by isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. It's also responsive to other outside metabolic reactions, and I just gave 2 examples. So that's the TCA cycle. Go over your notes, your PowerPoint notes more thoroughly. They have the mechanisms. This is just a brief overview with some of the important themes. And also I attempted to follow where the atoms are going. I sort of highlighted where the atoms are going so to sort of ease the transition of going, what is happening during what step? What is happening to the hydroxyl group? It's being oxidized, for example, or what is happening to the CO2? Okay, it's being decarboxylated. So I attempted to at least color code some of the important functional groups and their changes that occur. So this is the TCA cycle. The next step is to really determine what's going on with these NADHs and FADH2s that are produced. And also, we want to talk about the malate aspartate shuttle because that is directly involved in bringing in NAD plus or NADH from glycolysis into the mitochondrial matrix. Go over the PowerPoint notes, go over the lecture notes as well. So thank you.